All right, ladies and gentlemen, how's it going? This is going to be a 5-minute video on enzymes. So let's talk about enzymes. So enzymes are biological catalysts. In other words, they speed up chemical reactions. They help us with tons of reactions in our bodies, like so many reactions. But some really cool ones that you might want to know about, there's an enzyme called lipase that breaks down lipids, protease breaks down proteins, amylase breaks down certain sugars. Right, so they're used a lot in digestion, but a lot of other reactions as well. So enzymes lower activation energy, and you can kind of think of this like the amount of energy needed to start a reaction, okay? So there's this fun goat example. This rock's not going to do anything if the goat just looks at it. But if it gives it a little push, just like an enzyme does for a chemical reaction, then the rock is eventually going to fall off the cliff and the chemical reaction is going to happen. Right, so enzymes lower activation energy. This is a graph showing that. So think of a ball, all right, and you're trying to roll the ball up the hill. It's going to take a lot less energy for you to roll it up the dark green hill, right, which is the line with the enzyme, than it is to roll it up the light green hill, which is the line that doesn't have the enzyme. Now, once the ball gets to the top of the hill, it will roll down the hill on its own. All right, in other words, the chemical reaction will happen on its own. But to get a reaction to happen like this, you have to add activation energy. All right, so enzymes function. Enzymes either make or break things. They either build things or they break them apart. And how they do that is through the substrate binding to something called the active site. Right, and as you can see from this picture, the substrate fits perfectly to the active site. They're both complementary to each other. So much so that you can use the analogy of a lock and a key. So the key fits perfectly with the lock. And that's exactly how enzymes work. So let's take a look at this picture here. This is showing the enzyme substrate complex. In orange there, you have the substrate, in blue, you have the enzyme. And when they're together, they call it the complex. So pause the video. Is this enzyme making something or breaking something? It's actually breaking apart a disaccharide sucrose, di meaning two, into individual sugar monomers, fructose and glucose. Those guys there are monosaccharides. And it was done through a hydrolysis reaction. A lot of times hydrolysis breaks things apart, which when you add water, and then something called dehydration synthesis puts things together. That's higher level science than you need to know for this. Over here, we're having denaturing happening. So you can see that the triangular substrate is fitting into that triangular part of the enzyme there. But maybe it was heated up too much, or it was too acidic or basic, and now the substrate will not fit. All right, so pause the video again. Look at the bottom thing there. When its shape was changed, would the substrate molecule fit the bottom enzyme? No, it wouldn't have fit. All right, wouldn't have fit because the shape wasn't correct. Okay, so enzymes rely very heavily on structure so that the substrate is able to bind to it. Or here's just a graph. Okay, the graph is showing ideal conditions. For the enzyme on the left here, for temperature, it's probably about 40 degrees Celsius and 8 for the pH value. That would be ideal. All right, guys, nice work.